Today I'm going to be going over the Nirenberg and Mattaei experiments and the Nirenberg and letter experiments and how they relate to finding the genetic sequences that code for amino acids. First is the Nirenberg and Mattaei experiments that discovered that there were specific RNA sequences that coded for specific amino acids. The main components of this experiment were the cell-free environment, the poly-mRNA, and the 20 amino acids with one radioactive amino acid. The cell-free environment was obtained by breaking the cell wall and releasing the intracellular components. The remaining cytoplasm was used to synthesize proteins when mRNA was added. Nirenberg used E. coli bacteria as the source for the cytoplasm. The extract is placed in 20 test tubes, with each test tube containing a radioactive amino acid, 19 other normal amino acids, and poly-mRNA, which was mRNA containing only uracil units. The tube was incubated, and translation occurred within the test tube, resulting in a protein precipitate. The solution was filtered to separate the protein precipitate, which was checked for radioactivity to track if the radioactive amino acid had been precipitated. When phenylalanine was the radioactive amino acid, the precipitate was also found to be radioactive, indicating that the amino acid had been incorporated into the protein precipitate by the poly-mRNA. This indicated that the uracil chain poly-mRNA directed protein synthesis of phenylalanine and that mRNA transcribing genetic information from DNA coded for protein synthesis. Then there was the Nirenberg and letter experiment, which indicated the triplet nature of codons in the genetic code. Sequences of trinucleotides were used in this experiment. These sequences were synthesized mRNAs with codons that were long enough to attach to the ribosome and to be detected by radioactivity. Then there was a ribosome and 20 charged tRNAs with an amino acid, with one tRNA having a radioactive amino acid. After combining all components, the ribosome bound the mRNA and the corresponding tRNA with amino acid that matched the mRNA codon. In this case, the mRNA sequence is guanine uracil uracil, and the corresponding tRNA sequence is cytosine adenine adenine, with the amino acid being valine. The solution was then passed through a nitrocellulose filter to separate the unbound tRNAs in the solution from the tRNA and mRNA bound to the ribosome. The filter paper was tested for radioactivity to determine if the tRNA with the radioactive amino acid had bound to the ribosome. This method allowed the experimenter to determine which three nucleoside sequence coded for what amino acid. This verified the triplet nature of the genetic code and allowed the deciphering of the genetic code using codons.